connected, just to be ready to share your slides when it comes to your talk and then to unshare at the end. And I'll probably give you a bit of a warning with about a minute or so to go if you're starting to creep into time, but I'm hopeful we'll all go as planned. So we're on 12:30 now, so in the interest of time, I might kick off this next session, or sessions, I guess. So we're got a mix, I guess, of 2 sessions here with a series of talks, a bit of a mix of themes. Some will stay on the topic of the chemical origins of life, and we're going to end with this session with an invited talk by Dr. Surarajamani. But also intertwined in that, we're going to look at the modern analogues for primitive life on early Earth. And this one, I think, is highlighting a lot of the latest discoveries and technologies that we're seeing that really opens up what we know about different systems. And these are our windows to our past. And I deliberately chose this backdrop, which is an artist's impression of maybe what some of early Earth may look like, with some cool stromatolites foreground, which I work on over in Shark Bay, and some volcanoes doing their thing in the background. So we're going to hear a lot about different systems like these, but also on the very origins of life as well. So without any further ado, I'm just juggling 2 computers here, sorry. I'll kick off the first talk. Frankie Jutatas, apologies if I hope I've mispronounced your name, from Arizona State. And we're going to hear all about the oily ocean surface as a potential emergence of life. All right, take it away. Frankie, can you unmute?
Okay, I need to unmute. Okay, all right. Can everyone hear me?
Yep,
Hello?
all good.
Okay, all right. Thank you for the comment. Good day, everyone. It's I'm here in Arizona. So I'm Frankie. A little bit about myself. I'm a graduate student at Arizona State University. I actually did my bachelor and master's in aerospace engineering in Indonesia. So I'm pretty much familiar with this Pacific time and US time. So anyway, I work in a biophysics lab with Dr. Yurki and Dr. Haryadi. And one of the research questions that I'm interested in is the origins of life. So for the past 2 years, we have been working on these proposals that emphasize on the hydrodynamic forces in the origins of life. So by the end of this talk, I promise you that you will see how hydrodynamic forces can play a major role in the origins of life. But to justify this title, so let me define what I meant here by hydrodynamically active ocean surface as a cradle for the emergence of life. To do that, let's take a look at the next slide. So I believe some of us might have been familiar by petroleum productions in the oceans. So this is basically fossils, the organic molecules from fossil that are being buried and maturated by the high pressure and temperatures on the deep oceans, and it seeps to the ocean surface and creating this thick oil slick that we can see in Gulf of Mexico and some other place on the ocean. So here's the idea. So what we propose here is a similar mechanism could have occurred in the prebiotic world, but instead of using the organic molecules from the fossils, this process could have happened with the organic molecule from the micrometeorite. So what happened here is the oil slick that's produced by that process will have this organic ring, aromatic rings on its molecules, and it will create this oil slick that's sit on top of the ocean surfaces. So the advantage of having this model as the origins of life, because this oil slicks has density less than water and water insoluble, this model could minimize the dilutions and burial problems that we could see in other models of the origins of life. At the same time, the oceans, which in the first place produce this oil slick, would provide a sufficient geological time that allows the protozoals to evolve and migrate. I wrote here ocean gyre. So as we know, in oceans, we have this ocean gyre that can accumulate these organic molecules. So during this modeling process, we did a lot of calculations and verification, some debates between me and Dr. Yurki and Dr. Haryadi to convince ourselves how this model would be sufficient. But I will just mention two reasons why petroleum productions in a prebiotic world would be more efficient than the biogenic petroleum that is produced from the fossils. So one is there is no biological activities such as microbes that can decompose this petroleum. And secondly, the young Earth has these larger temperature gradients within its crust, which allow these petroleum maturations to occur closer to the ocean surface, which essentially increase the yield of these oil slick productions. So now that we have these convincing models of how the oil slick could have occurred in the prebiotic world, the next question is to look at how protocells could have been produced in this model. So as we know, ocean surface is an aggressive and a very active environment. There are two major forces of, there are two major phenomena that cause hydrodynamic forces. One is the breaking wave, and another one is if you look at the raindrop that could occur on the oceans, we think that this raindrop could perturb this oil slick, and the surface tension forces will create this oil droplet on the ocean surface. So here's what makes it more interesting. So instead of looking at the hydrodynamic forces that play a role on the birth of these protocells, we think that these hydrodynamic forces will also contribute to the other process of this protocells model, namely the growth, replications, and the death, which is induced by the burial of this oil droplet. So this idea is not new. In 1924, Oparin proposed that quartz sulfate, or the first replicators, use similar mechanisms to replicate. In these proposals, we propose oil droplet instead of quartz sulfate as these first replicators by looking at the astrophysics and geophysics model that is plausible in the prebiotic world. So anyway, now that we have this model, we want to test it in the lab to convince ourselves whether these hydrodynamic forces could have done all of these processes for this protocells model. So let's start with the bird. So we built this a raindrop simulator in the lab. So all the parameters are being adjusted to prebiotic conditions. So like the raindrop diameters, the impact speed is adjusted to the prebiotic atmosphere. So this is the system. So we have water representing the ocean. And we have this oil slick represented by this simple oleic acids. And we have this surfactants molecule dissolved in the water solutions to stabilize any protocells that could have been formed. So what's interesting is when we apply these hydrodynamic forces via a raindrop impact, we found that not only oil droplet consisting of oil material could be produced, like this one, but we could also see some oil droplet that consists of water liquid that encapsulated by this shell of oil, which mimic something like vesicle-like structures. So this is the distributions of this droplet. It follows this by exponential distributions. The majority of the size of this droplet is about twice the size of modern plant cells. We have a model that basically analyzed how this droplet size could have survived in the ocean environment, having those gyres and so on and so forth. I'm not going to talk the details about that in this occasion, since we have limited time. So let's move on into the replications, which basically how the parents make children and transmitting information, allowing Darwinian evolutions. So since this oil droplet is this oil droplet doesn't have a molecular machinery, it is intuitive to think that it must have relied on external forces to help them in order to do replications. as eager as I am to show you, demonstrate the replications of oil droplet, I haven't been able to do so yet. So what I'm going to show you here is the primitive replications. In this case, is the fragmentations. So we have this about 2 millimeter of oil droplet sitting on a water body. This is inside a beaker. And we apply just a single raindrop impact. And we can see here as the raindrop tear apart this oil droplet, the surface tension forces quickly encapsulate this droplet, producing this sort of daughters of oil droplets. So this is the replications, and let's see the growth, how hydrodynamic forces could have contribute in the growth of this oil droplet. But before jumping to the growth, while we try to answer these questions and addressing this this problem, we look again into this model and we ask questions to ourselves. If we have these light organic molecules sitting on top of the ocean surface, some of these organic molecules, it has some high vapor pressure. So we thought that, okay, if we have some high vapor pressure, could it be that some of this light organics molecule evaporates and the remains will turn into a tar ball? So to address this concern, we collected a tar ball sample from Galveston in Texas. So this tar ball is a spongy rock that you could find on the ocean surface. It looks heavy, but it's actually not. This was taken during a GRC Origins of Life earlier this year, before this COVID outbreak was detected in the US. So here's what we did. So we fragment this tar ball sample. We put it in an airtight chamber. We have a xylene here, xylene solutions. So the questions that we tried to ask is, if we have free molecules of xylenes on the air, whether or not these free molecules of light organics could turn this tar ball into the oil slick, which will prevent.
Frankie, one minute.
Okay, all right. So as you can see here, this is the image, and we can see how the oil slick could have been produced by the process. And but what's interesting is when we do a weight measurement, we can see here there is a weight gain for all these samples. And you can see it has a different rate, some of them, which indicates inhomogeneity of this sample. But the takeaway that we can see here, there is a weight gains, which basically means that if there is organism, it can take external nutrients to its body. So I will end this talk with a conclusion. So we show here a proposal that can circumvent the dilutions and burial problem by providing sufficient geological time. And as promised, all of us see how hydrodynamic forces could play a role on protocell models, in this case, the oil droplets. So with that, I would want to thank the lab that I'm working at and artists that helped me with all those illustrations. Yeah, with that, I'll be happy to take any questions.
Thank you, Frankie. That's a really good talk. Everyone loves the raindrop simulator, I think.
Thank you.
Good time for just maybe a couple of quick questions. One from Luke. I don't know if you can see in the chat here about the benefits of organic surface slicks applicable to all surface water systems or just like a salty ocean.
Can you repeat that question? How to see that question on the chat? Is it on the chat?
Just the idea of whether the benefits of an organic surface slick is applicable to all surface water systems such as hot springs, etc., or are the aspects unique to salty oceans, you think?
Oh, I think it's not unique to salty oceans. I think it could also benefit from other habitats of the origins of life. We haven't tested yet the interactions between these organic molecules with salty environments. I believe it will change the surface tensions and perhaps some of the interactions to the organic molecule. That will be interesting to see in other environment as well.
Sure, no, that's really good.
Okay.
We might move on just for sake of time, but there's a couple of other questions in the chat you might want to have a look at if you get a chance, Frankie, and we can kind of keep the conversation going. Thanks.
All
so
right, thank you, everyone.
Thanks very much. If you stop sharing your screen, then we'll move on to the next. So Siddharth Pandey, if you're there, could you jump on? How you going? Great. So Siddharth from Amity University in Mumbai, going to tell us all about Indian terrestrial analogs. Looking forward to it. Take it away, mate.
Thank you. My screen visible?
Yep. So you can just, yep, that's great. Off you go.
Thank you. Warm greetings to everybody. I'm here in Mumbai. My name is Dr. Siddharth Pandey. I'm heading a new center of excellence in astrobiology that we have established just this last year. But the work that we have been doing in India goes back to 2016. And this image that you see in the backdrop is actually from a place called Ladakh. So Ladakh has been kind of, you can say, a meeting ground for many of us here who kind of kickstarted our work to work together in this field and kind of assemble ourselves and form an astrobiology community. So this picture is actually quite, you can say, memorable for us since we started work. And this new center is mainly looking at early Mars analogs. So my abstract mentioned Mars missions as well as icy moon missions, but I will limit myself to just 2 analog sites that we have identified. One of them we've started doing some work, and then the other one we have identified what work we need to do via several scouting trips. So this talk is an overview talk to give you a general broad picture idea, and hopefully if there's questions, I can take them subsequently on the work that needs to be done further. So just to give you a perspective of India, things are getting quite exciting here. The space agency has now got ambitious plans to send humans into space, into low Earth orbit. That's become the national priority with the Gaganyaan human spaceflight mission, as well as on the space science things. India has launched 2 lunar missions, and then the third one is slated to be launched early next year to be a rover lander that will be arriving on the south pole of the moon. And these are all obviously critical technologies that are stepping stones towards conducting more work on the Martian surface and obviously leading up to astrobiology. So all the critical elements of a rover, drilling on the surface, ISRU, communications, all of those things need to be tested. And that's exactly what analog environments are for. So we already know this, but just to reiterate the importance of an analog site towards development of missions. We've seen this happen, in the US, in Europe, and also now we're trying to understand ways in which we can incorporate analog research that normally serves the purpose of astrobiologists, but also better understand ways in which we can test technologies and instruments, in the field, as is being done by NASA, ESA, and some of the other agencies. How exactly we can do that, for the Indian space experiments that have to be packaged for lunar as well as future Mars missions. So I present to you Ladakh. Ladakh is a site in northern India. It's in the northernmost union territory of the country. And this particular site has been classified as a high-value analog site for science, technology, and operations testing by several members of the astrobiology as well as Mars analog research community. And Ladakh is basically a high-altitude, cold, dry desert environment, which is obviously exposed to high UV radiation, low oxygen levels, and several features, unique features, which I'll talk about in a minute, that are giving us you can say a look into what ancient Mars would have been like when it was warmer and had liquid water on its surface. And also there is the diversity of different environments there that exist. So since 2016, I have been fortunate to work alongside several astrobiologists from Australia, New Zealand, the US, Europe, and also in India to work together in the field as part of the Spaceward Bound India program. And there were several things that we did there. One was obviously looking at all of these different regions. Because it was a scouting trip, there was a lot of initial groundwork that had been done by Earth scientists, but we were there with a specific focus in understanding, what's the relevance from a Mars exploration perspective. So I've tried to summarize things, so I'm going to keep things, really, really short here, but basically to give you a gist of the different kinds of environments. So you're going from 3,500 meters above sea level all the way up to about 5,600 meters sea level. So within that altitude range, what is the variation in the topography, what is the variation in the diversity of microbes that are found in these different distinct environments. So we were looking at meltwater pools, glacial sediments. There's also sand dunes that exist, similar to the ones that we find inside craters on Mars, and also ephemeral ponds inside them, which have clay deposits. Looking at high altitude hot springs, several of these hot springs are, hot spring fields rather, are exciting because they contain boron, and that's a critical element that is connecting us to all the different kinds of terrestrial hot springs we have found in Pilbara, for example. So Dr. Martin van Kranendonk and Luke and others are working alongside astrobiologist Sudha and Dr. Sudha Rajamani and Dr. Mukund Sharma here in India, and others in the US as well now, in trying to identify ways in which we can connect the dots in identifying how terrestrial hot springs were perhaps one of the places where life could have potentially started on this planet. There's also saline lakes and shorelines. So several of these lakes, or rather wet basins now, are demonstrating how over hundreds and thousands of years the landscape has evolved, and how we're seeing the receding shorelines, how water is evaporating, and how we find communities of microbes that are inside the lake as well as on the shorelines, and the unusually high biodiversity that is demonstrated there. And on some portions of the lake shores, we also find these permafrost mounds, and our team was also able to excavate and confirm the presence of permafrost. And there's obviously a seasonal variation, and we are seeing a lot of visible effect of climate change at these particular sites. Either that might be the pH, salinity, or the soil carbon content that has been changing. In fact, just over the last 4 years, we've actually seen visible changes. So we are actually planning towards a trip next year in 2021, where we can continue on with further exploration of these particular sites. And I think the takeaway from Ladakh is that within about a 50-kilometer radius, you have all of these different, very diverse features. And that allows scientists to test their theories in different environments, whether they're looking at hot spring sediments or they're looking at saline sediments. So really a little bit for everyone to come there and test. And myself, from an engineering background, I'm obviously interested in seeing ways in which we can test the technologies. And one of the systems that was actually tested there was part of the ExoMars 2022 mission, an instrument called HABIT, which was tested. And HABIT mainly looked at firstly, obviously measuring the ultraviolet radiation, local wind speeds, relative humidity, but mainly looking at the ability of salt to get hydrated in that kind of environment. And it was basically testing the rate at which deliquescence was taking place. So it was a very neat experiment that we tested alongside other analog sites, I think in Iceland and in Sweden, that they conducted those tests, and they were able to validate some of their performance parameters in the field. So just a table of different activities that we conducted. So everything ranging from exercising a new type of landform mapping called regolith landform mapping that allows us to do ground truthing of surface features by comparing maps that have been created by airborne as well as spaceborne data, and looking at ground-based measurements taken from, for example, a rover or an astronaut on Mars surface, and then basically breaking down that map and identifying different features. So that's a new technique that Dr. Jonathan Clark had led. So we did that for some of the glacial sediment sites. Obviously, a lot of parallels in geomorphology, looking at the different features and how they have evolved over time. Because of its remote and very pristine nature, the sites have only seen very little human intervention, and most of the weathering that has taken place, the impact of the atmosphere and the landscape and how that has evolved, looking at the wind patterns and seeing how the terrain has evolved, that's something that's very relevant for people who are looking at understanding topological feature evolution on Mars. Instrument testing I already spoke about. We used two field-based techniques, ATP and LPS, for measuring biomass distribution across the entire transit that we covered in Ladakh, and we were able to find regions which obviously had a larger distribution. Specifically the dune pond sites with the clay deposits, as well as around the saline shore lakes. And all the details are there in the paper. I'm happy to share the link that came out last year that gives an overview of the work. And then there's obviously topic-wise papers that have also come out, and Dr. Sudha Rajamani, I'm sure, will be talking a bit about that as well. Quickly moving gears to our second site, which is Lonar crater. So on the lower right side of the screen, you're seeing the map of India and one of the largest flood basalt plains in the world, the Deccan Flats. We have a crater site that was formed about 500,000 years ago when a crater struck and caused a roughly 2-kilometer wide, very well preserved crater in basalt. And that's exactly what makes it unique. Being a basalt impact crater, there's just a few, 2 or 3 of them in the world, and Lonar is quite accessible, and it's a direct analog to, in our opinion, to Jezero crater on Mars, which is where the NASA Perseverance rover is going with a very strong astrobiology, objective. So we've drawn parallels between the 2 craters, and we made 3 trips last year, and then we plan to go back again. We are mainly interested in trying to understand what are the morphological comparisons of the delta gullies inside. So basically both Lonar and Jezero have a delta, an inlet, and then Lonar, obviously today being a modern analog, has an active lake system within it. And we see that from Jezero that, of course, at one point of time, it also had a large standing water body inside it. How exactly was the water flow affecting the mineral alteration in the gullies? So we've basically divided the crater into 3 different, you can call them planetary analog crater terrains. So 3 different pacts. So one is the sediments inside the delta fan. The second is the inner crater gullies. And the third is the crater ejector blanket and the gullies that are associated with it. So we have divided it into these 3 different terrains. And for each of them, we are basically now starting work in understanding what exactly are the morphological comparisons, the geochemical, mineralogical, and the composition of organic distribution, in those different terrains, and how exactly, what implications do this distribution of minerals and their alteration have, and how does it help us understand the subsurface habitability potential of the site? So we're not obviously interested in the current modern foliage and the fauna that you see, the flora that you see there, but mainly in the ability to trap these ancient biological signatures in there. So that's a starting point for us, and we hope to do more campaigns. I'm guessing not this year with the current situation, but perhaps early next year or later part of 2021, where we're basically looking at drone-based capabilities in which we can test some of the hyperspectral data that we can capture, and then see how useful what can we really do with drones in such a terrain that's not easy to traverse for rovers or humans. So that's one focus of this particular group. So that's, I think, pretty much it. I didn't want to talk too much about the other sites, but the third site is Kutch. Do you have a minute?
Yeah, just 1 minute, thanks.
Okay, thank you. So Kutch is the other site, which Dr. Michael Macy, in fact, visited earlier this year from Open University. And there's several people who are already working in that region. People have been studying the jarosite occurrence that has been found in Kutch, which is obviously a very relevant mineral structure that we find on Mars as well. We're also interested in, obviously, some of these seasonal ponds that tend to form inside these large salt flats, and then looking at the salt-loving life that exists in there. And there's actually a talk that Dr. Anushri is giving on Sunday on that, which is part of our center webinar as well. So I'll be happy to share the link for people who are interested in understanding more about Kutch. And finally, hopefully, I'll be able to give a presentation, perhaps next year or maybe 2 years from now, on some of the sites in the ice-free regions of Antarctica, where India actually has a station. And there's several outcrops that are exposed, ice-free completely, and these pristine ponds, which actually have microbial pinnacles that have been found at the pond bottoms. So there's several of these sites, and there's obviously more to be done. For Ladakh specifically, I invite researchers who are listening to me to reach out to me who are interested to do work there. Amity is working towards establishing a research station to start with a 3-month semi-permanent structure that will be placed that will support scientists who will be working in that particular area, and also for Lonar. So I think that's it from me, and I'm happy to take questions and take things forward. Thank you.
Thank you, Siddharth. Fantastic. Some really, really cool images you got there. It's beautiful. Yeah, we don't have time for too many questions. Quick one, which I was also thinking of myself. Do any stromatolites up there in Ladakh? Or?
So there have been, I think, this needs to be further investigated. We have actually taken images of microbial pinnacle structures inside the hot spring little ponds that form, but there hasn't been any documented evidence of finding stromatolites up there, at least to my knowledge.
No worries. That's okay. All right, we might just move on, and there might be some other questions in the chat which you can always get to if needed. But thank you, thanks again.
Thank you, Brennan.
So if you want to stop sharing your screen, we'll move on to the next speaker. So this is the first of two speakers actually from my group. Moving into more modern analogues, focusing on my stomping ground, which is Shark Bay in Western Australia. And the first is by my student, currently now postdoc, Hon Lung Wong. He started with me as an honours student, his PhD, and he's in a few weeks when they actually open the borders, is going to head off to a postdoc in Europe. So, and he's just going to talk on one aspect of his PhD in this talk, everything about dark matter. So Alan, if you want to jump in and share your slide.
Yeah, sure.
If you want to start sharing slides. So we're going to hear all about dark matter, but not the dark matter that you probably are thinking of. Microbial dark matter.
Can everyone see this?
Yeah, so if you want to just, yep, that's it. That's excellent. All right. Okay.
Yeah. Okay. Hi, good afternoon, everyone. So my name is Alan Wong, and I am working in Brendan Burns Lab at UNSW. And now I'm going to talk about microbial dark matter matters in microbial mats. So I will start off with introducing what are microbial mats. So microbial mats are organosedimentary structures that usually have very steep gradients of various nutrients. So indicating a very high number of microbial interactions and putative metabolic niches at a millimeter scale. And microbial mats were dominant on early Earth, and the modern microbial mats are a great analog for ancient stromatolite systems. And one of the most importantly, microbial mats are suggested at one of the places to harbor the earliest eukaryotic cells on Earth. So one of the places that harbors the most extensive microbial mat systems is Shark Bay. So Shark Bay is located in Western Australia, and it has an extreme environment by having high salt concentration, high UV radiation, and it's oligotrophic, meaning that there is low nutrient concentrations in Shark Bay. And it's similar to Precambrian stromatolites. It is also a World Heritage Site, so it's under protection. And here is a photo of two types of microbial mats and stromatolites. So in my work focuses on smooth mats circled here because of higher microbial diversity and more novel findings in smooth mats. So I'll talk about the previous works on Shark Bay microbial mats. So we have elucidated the bacterial and archaeal communities, and we have proposed putative niches in microbial mats. We have done metagenomics to look into the functional genes and putative ecological roles of the microbial communities in microbial mats. And one of the interesting findings is that there are a high number of novel bacteria and novel archaea that are affiliated to microbial dark matter. So what is microbial dark matter? There are microorganisms that have no cultivated representatives. So usually they have unknown genomic content, and we don't know what they are doing during the microbial mats. We don't know their putative niches or functional roles in the Shark Bay microbial mat systems. And previous studies on microbial dark matter are usually recovered from underground water, and it is suggested that there are early branching microorganisms. In other words, they resemble microorganisms on early Earth. So one of the ways to look into microbial dark matter will be metagenomics. So metagenomics provide us with a very powerful DNA sequencing platform that provides us with high resolution of community DNA, and it can detect the genomic contents of the MDM in Shark Bay microbial mats. So in my work, I reconstructed individual MDM bacterial and archaeal genomes so that I can look into their genomic content. And so here's my aim in this project is to investigate whether microbial dark matter in microbial mats play any roles in nutrient cycling and to look into their genomic content. So here is a schematic of the metagenomics workflow. So the main take-home message here is that I used metagenomics and I reconstructed genomes or MAGs affiliated to microbial dark matter, and this will form the basis of the results that I'm going to present. So here's the one of the results of microbial dark matter in SharkBay. So here you can see a phylogenetic tree of 115 reconstructed MDM. I will just refer microbial dark matter as MDM throughout the talk. So here is I've reconstructed 115 MDM genomes. And here you can see the circle dots here. They represent the presence of certain metabolic pathways. So MDM are classified into 10 groups. And these 4 groups here in red are the ones that have little or even the absence of metabolic pathways, indicating that they have minimum or limited metabolic capacity. So I also looked into, due to their limited metabolic capacities, I looked into the carbohydrate enzymes in order to see what kind of carbohydrates that they can degrade. So the figure here, y-axis here, represent different types of MDM genomes, and the x-axis here indicates the different types of carbohydrate enzymes. And what I found is that they are able to degrade a wide range of carbohydrate enzymes, including starch, cellulose, chitin, and various animal and plant polysaccharides. And I also found that they encode for a wide range of hydrogenase. There's up to 16 types of hydrogenase in the MDM genomes, despite the limited metabolic capacities. And this suggests that they may have a role in SharkBay microbial mats for anoxic carbon transformation and hydrogen turnover. And here are some illustrations of the reconstructed genomes of the MDM microbial dark matter in SharkBay. And I found that they have very high capacity of hydrogen production, usually related to fermentation. And the genomes also encode a high abundance of carbon monoxide dehydrogenase that can oxidize carbon monoxide to carbon dioxide. And they mainly digest peptides and carbohydrates. And one of the very interesting findings is the eukaryotic signature proteins that were found exclusively in eukaryotes before, but now it was identified in Ascalachia. And my colleague Fraser will be giving you more details in the next talk. So due to the limited metabolic capacities found in MDM in SharkBay, it led me to think that they may be, they are likely host dependent and may be parasites in order to obtain energy or nutrients from their host. So I looked for diversity generating retroelements, DGRs, in the MDM genomes. So DGRs are fast evolving proteins that allows microorganisms to attach to the surface of their host in order to obtain nutrients. And over half of the MDM genomes encode for DGRs, indicating that they are likely host dependent. And one interesting finding is a novel rhodopsin called schizorodopsin, and they were found in a wide range of MDM genomes in SharkBay. It was only found in Ascalachia from a paper last year, but in SharkBay microbial mats, we have expanded the phyla that encode for schizorodopsins, including Deepanarchaea, Tacharchaea, and Parcobacteria. It was not until earlier this year that schizorodopsins were found to have a function of a light-driven hydrogen ion pump, but there's still inconclusive function of schizorodopsin. So it can be only suggested that schizorodopsins enable MDM in SharkBay for light sensitivity. And another interesting finding is a range of rubiscotes found in microbial dark matter. These types of rubiscotes allow MDM to break down nucleotides into ribose. And the co-occurrence of these two genes here, AMP phosphorylase and ribose phosphate isomerase, suggests that while MDM are breaking down nucleotides into ribose, they can also fix carbon. They can also undergo carbon fixation at the same time. And this gene may be important to MDM in shark bait, as they can break down nucleotides to ribose, which can in turn be fed into the central carbon metabolism, given the oligotrophic environment in shark bait and the limited metabolic capacity in MDM. Rubiscome may act to supplement carbon intake for the MDM genomes in shark bait. So we found a wide range of hydrogenases and a high abundance of carbon monoxide dehydrogenase in shark bait, MDM, and also various types of rubisco that can break down nucleotides to ribose, putatively fixing carbon in the process. So it is suggested that hydrogen, ribose, carbon monoxide, and carbon dioxide are the main energy currencies for shark bait microbial dark matter. And here's a summary of the project. So there's a large abundance of microbial dark matter in shark bait microbial mats, and they all have minimum metabolic capacity, mostly breaking down carbon, and may have a role in anoxic carbon transformation and hydrogen turnover. They are likely host dependent, and they encode for schizorodopsins that suggest for light sensitivity. And microbial dark matter in shark bait likely use hydrogen, carbon monoxide, and carbon dioxide and ribose as main energy currencies. And this may infer to microbial lifestyles in the Precambrian period. So here's a ecological model of what I've been building for the past several years. And I've incorporated the ecological and functional roles of MDM in this model. So this is a ecological model of shark bait microbial mats. And if you would like more details, feel free to contact me, or maybe read this paper that got just accepted a few weeks ago. So thank you for Brennan and my colleagues that helped me along this journey. And this is the end of my presentation. And do you have any questions?
Excellent. Thank you, Alan. Great journey you have there. Any questions from people either in the chat, or if people wanted to unmute their mics, they're more than welcome to ask Alan a question. We've got a couple of minutes. What are your thoughts, Alan, on being able to get more of these in culture based on all of your data? Do you think there's more of a chance you're about to get some of these unculturables back into culture?
Oh yeah. In fact, some of the Ascalacea were brought into culture earlier this year. It was published, and my colleague Fraser will be working on cultivating them. So now we have the genomic contents of the microbial dark matter. So we know what kind of nutrients they need, or what kind of conditions they might require to grow. And we'll be trying to grow the cultivate the MDM in the projects afterwards.
Yep, oh, that's good. There's a few questions in the chat which you can have a look at if you want a little bit later. We might move on just because of time to the next talk. So thank you again, Alan. If you want to unshare your screen, and then we'll move on to your colleague in crime, Fraser. He can start sharing his screen. You're going to tell us all about Asgard-Archaea as potentially our great ancestors starting the whole eukaryotic chain of events. All right, Fraser, the floor is yours.
Hi everyone, can you all see my screen?
Yep.
Perfect, thank you. Okay, hello everyone. Welcome to my talk. My name is Fraser McLeod and I'm from the Burns Lab at the University of New South Wales. Before I begin my talk, I'd like to acknowledge the Indigenous custodians of the land, the Gadigal people of the Eora Nation, and recognise their connection to the land, water and culture. And I would like to pay my respects to the elders past, present and emerging. And also much of my research was conducted in Shark Bay or Gufragudu, two waters. And so I would also like to acknowledge the rich Aboriginal history of many cultures in Shark Bay. I'm going to be presenting today on my favourite group of microorganisms, the Asgardarchaea, or the Asgardians of the galaxy, the most complex known group of archaea, which have some really important evolutionary implications that are rewriting the textbooks and really changing how we understand how life evolved. So approximately 2 billion years ago, life on Earth underwent a dramatic increase in cellular complexity. The eukaryotic cell from which we are all descended emerged from what is believed to be the fusion of the two existing cell types at the time, the bacteria and the archaea, either in this kind of dual fusion model where one cell was engulfed by another, or a kind of tripartite fusion of multiple cell types where multiple cells were engulfed and became the membranes of eukaryotic cells. And so what's really shaken up our understanding of eukaryogenesis and the role of archaea in eukaryogenesis was the discovery of the Asgardarchaea a couple of years ago. The Asgardarchaea are a superphylum of archaea named after the realm of the gods in Norse mythology, or maybe they were just Marvel fans. And so far there has been 5 phyla of Asgardarchaea that have been described, and these include the Loki Archaeota, the Odin Archaeota, the Heimdall Archaeota, the Hel Archaeota, and the Thor Archaeota. And what's really interesting about these Asgardarchaea is when they're included in phylogenetic analyses of the tree of life, eukaryotes either branch from within the archaea, either as a group of the Asgardarchaea or as a closely related sister group to it. And these Asgardarchaea also encode a number of eukaryotic-like characteristics, which have led a lot of groups to suggest that they were one of the cells that was involved in the origin of eukaryotes, often suggesting that they were the host cell. And so I'd just like to quickly introduce microbial mats and stromatolites, which are the environment that I study. And these are these layered communities of microorganisms that date back approximately 3.5 billion years. What's really interesting about these structures is that they peaked in abundance coinciding with the rise of eukaryotes. Sadly, they've decreased in abundance more recently due to the rise of burrowing eukaryotes. But they still remain present in isolated environments, including the saline waters of Shark Bay in Western Australia. What's really cool about these microbial mats, and one of the things that really help, one of the reasons I think they might be involved in eukaryogenesis, is the sharp change in oxygen levels for the microbial mat leads to layers of dramatically changing redox potential, which allows organisms that wouldn't normally be able to interact together to interact with each other. And one thing that's been suggested about these microbial mats is they might be an environment in which Asgardarchaea could interact with delta proteobacteria and alpha proteobacteria in the origin of the eukaryotic cell. And so one of the things about microbial mats is that Asgardarchaea previously hadn't been identified them until Alan managed to recover some genomes of Loki Archaeota and Thor Archaeota in some microbial mats, and more recently Heimdall Archaeota. And so I revisited these structures and found that really Asgardarchaea were the most abundant group of archaea in these structures. They reached up to about 5% of the total cells, or 60% of the archaeal diversity. And there was a really high abundance of Loki Archaeota and Thor Archaeota, while Heimdall Archaeota and Odin Archaeota were more in the rare biosphere, so we're talking on the less than 1,000 cells. There was also a small amount of unclassified Asgardarchaea, suggesting there are more groups of the elusive Asgards yet to be discovered. Sadly, I wasn't able to get enough information to build genomes of these, but fingers crossed. So I've really had three approaches in the way that I've approached my research into the Asgard Archaea. Firstly, I've been doing some imaging to understand the morphology of the Asgard Archaea. I've been doing some genomic characterizations of the Asgard Archaea, and lots of phylogenetic approaches to detect different microbial interactions between the Asgard Archaea and other groups. And really what I want to do is I want to combine these different forms of evidence to propose new models about how eukaryotes evolved. And that's something I'll come to towards the end of my talk. So I've been taking some fluorescent in situ hybridization images and some live images of these Asgard Archaea. And I found these generally small cells where, really interestingly, if you look at this blue channel, this is the DNA, and the DNA is really localized within these cell structures, which is indicative of a eukaryotic-like cellular organization. I won't suggest that this is a nucleus level of DNA organization, but one thing I've suspected is it might be some sort of nucleosomal organization, because these Asgard Archaea encode for a number of eukaryotic-like histones and similar processes, so they might have a different way of organizing their DNA. I also found I took these live images of Heimdall Archaeota, and I found these larger cells of Heimdall Archaeota in oxygenic environments. Increasing oxygen seemed to produce different morphologies, and this kind of DNA localization was also maintained in these larger cells. And so one of the things that Alan alluded to earlier was that I detected these eukaryotic signature proteins in Asgard Archaea, and these are proteins that didn't normally have homology in the other groups of life, the archaea and the bacteria, purely eukaryotic-like innovations. And they were originally used as an argument that there wasn't an evolutionary relationship between eukaryotes and archaea, and that the eukaryotes were their own group. What I found when I looked at them is I found these eukaryotic signature proteins that had previously been discovered in Asgard Archaea, so including an actin-based cytoskeleton, some membrane trafficking machinery, and a ubiquitin system. And I expanded the eukaryotic-like systems that we have in Asgard Archaea to include a lot of eukaryotic-like translational machinery, proteasomal and signalosomal machinery, a bunch of eukaryotic-like motor proteins that probably interact with the actin-based cytoskeleton, an expansion of these GTPases, which control and regulate a bunch of eukaryotic-like processes, N-linked glycosylation system, which is the main post-translational modifications you find in eukaryotes. And really the message I want you to take away from this is that a lot of the unique processes that eukaryotes possess actually likely their origin in an ancient archaeal system. Now that we're starting to study the archaeal origins of the eukaryotes, I wanted to understand which groups these organisms were interacting with and see if they had any syntrophic relationships that could have led to the rise of eukaryotes. And what I found is I built this horizontal gene transfer network where I assessed the phylogenetic placement of different genes in the genomes of Asgard Archaea and other groups in microbial mats, and found that they'd exchanged a lot of genes with sulfate-reducing delta proteobacteria and sulfate-oxidizing alpha proteobacteria. And if you remember what I mentioned earlier in the talk, these are some of the cells that have been suggested in the past to be involved in eukaryotes. And so I assessed the metabolisms of different members of the Asgard Archaea to look at what they all had in common, so we can infer from the common ancestor of Asgard Archaea and eukaryotes what kind of metabolisms it might have possessed, and what kind of metabolisms might have led to the origin of eukaryotes. I found a lot of cool pathways involved in carbon, sulfur, and nitrogen cycling that suggest that the Asgard Archaea might have very important roles in global cycling of these nutrients. But one thing I really found that was really important is a lot of Asgard's archaeal metabolism was based on the cycling of hydrogen and acetate. So acetate being produced through the Wood-Lundell pathway. And then the system of schizorodopsins pumping protons inside an Asgard Archaea. And then these protons being converted into molecular hydrogen by these group 4 nickel ion hydrogenases. I think this is likely coupled to ferredoxin reoxidation, which acts as a terminal electron acceptor for these Asgard Archaea. But when it comes to the syntrophy between Asgard Archaea and other groups, I think that this hydrogen and the acetate produced by the Wood-Lundell pathway could be exchanged and fed off by a bacterial partner, maybe a delta proteobacteria. The thing about hydrogen metabolism is as hydrogen is produced, it actually inhibits enzymes that would be producing the hydrogen. And so you often need a partner organism to mop up this residual hydrogen. And the delta proteobacteria might have also been able to feed off the acetate and the formates that were produced as in excess from the Wood-Lundell pathway. And so what I think is also going on is that the delta proteobacteria is interacting with the alpha proteobacteria in this cycling of sulfates. So you have sulfate reduction being performed by the delta proteobacteria and sulfate oxidation performed by the alpha proteobacteria. So they're interacting together. They're both feeding off the acetate and the formate and the hydrogen. And they're also mopping up the oxygen, which would be toxic to the Asgard archaea. To allow the Asgard archaea to occupy more aerobic niches, these cells might have formed an intimate relationship where the delta proteobacteria and the alpha proteobacteria protect the Asgard archaea from oxygen. This eventually led to the fusion of these cells through an engulfment by the delta proteobacteria, which later led to this eukaryotic cell. You have the DNA localization inside the Asgard archaea produce the nucleosome. The Asgard archaeal cell produces the eukaryotic nucleus. The alpha proteobacteria evolved into the mine, and then the delta proteobacteria became the eukaryotic-like host cell. That's my metabolic intimacy model for the origin of the eukaryotic cell. I would like to thank everyone who's worked with me over the years, and especially my supervisor, Brendan Burns, and my mentor and my brother, Alan Wong. Without the two of you, I would be lost in the void of research, wandering aimlessly. So thank you very much.
Excellent, thank you, Fraser. Research is all about being lost and wandering aimlessly, so don't worry about that. Excellent talk, love the Asgards. There's a couple of questions. I'm going to start off with one. So we've just, and Alan, and talk about what we're finding in the mats and then the Asgards potentially as a link. Is this a huge bow to draw that maybe this, if there was a fusion or a symbiosis, that it could actually have occurred in an ancient microbial mat, or is that a long bow to draw?
I think the microbial mats are the most likely environment for this fusion to take place, really because they have these sharp redox gradients where something like an anaerobic Asgard archaea can actually be brought into really close proximity, because it allows the fusion of these cells that would very rarely have an association together, and allows them to interact. I think microbial mats would be the best environment for eukaryotes to emerge.
I'm not sure if it was just me just cutting out. That's excellent. There's just one quick question from Shabani. Just whether you have a chance to explain a bit more on the syntropy. So how did, very briefly, the methods you used to determine potential syntropy?
Yeah, so I basically broke down the genes that were found in the Asgard archaea and the alpha proteobacteria and put them through this phylogenetic pathway, which detects horizontal gene transfer events. So if one a gene in an Asgard archaea phylogenetically looks like it might have emerged from alpha proteobacterial systems, then that is detected as a syntrophic relationship. And so I looked at when the groups which were well above the average of normal levels of transfer, and interpreted that as an indication of syntrophy. Very theoretical, but there has been some research recently that shows that horizontal gene transfer is one of the most effective ways of picking up syntrophic relationships, and it's much less likely to give you a false positive correlation analysis do.
There may be some issues with Brendan's connection, so I'm just going to pop in and ask. Actually, no. So Fraser, you'd have more questions in the chat, so please head there to start answering them. Thank you so much for that great talk.
Thank you for having me.
Yeah, my
Brendan, you're
connection's
back?
timing out here. Yeah, but my Zoom connection is playing up. I don't know if I'm on or not. I can't tell.
Yeah, you're
If you can hear me, that's the main thing, and I'll try and. I can't see anything, but I can. If you can hear me, then we're gonna go to the next speaker, which is Harold Bernhardt. If you can share your screen, Harold, I'm gonna try and get back into Zoom. I'm having problems. Can you hear me okay, Anna?
Yes.
Okay, cool.
And Harold's on as well.
Excellent, Harold. So I don't have your affiliation here, Harold, so I can let you introduce yourself, but we're going to go back into the maybe messy chemistry side now, talking about amino acid activation. Hopefully I can jump back in. All right, I'll let you take it away, Harold.
Okay. Is that working?
It might just take a moment. Right now, we don't see your screen just yet.
I pushed the share screen button, but I don't know if that's my computer's going a bit slow at the moment as well.
Okay, so after you pick the button, you have to select the window that you want to share.
There's always a catch, isn't there? Okay.
Yeah. So if you don't see that window, just browse. I mean, look around a bit and find that.
Look around. Sorry, look around.
Down the bottom.
Oh, okay. Hold on.
Screen down the bottom.
Screen down the bottom.
Yep, that should be it. I hope.
Sorry, I've never shared a screen on Zoom. I've done lots of Zooms before, but not a So that's still. I take it that's still not showing.
That's right, it's still not showing. So down the bottom of the Zoom screen, there's a toolbar. There's a green button that says share screen. After you click that button, another window pops up.
Oh, okay.
And that window is inviting you to choose which screen you want to share. Like, do you want to share a whole desktop or like a browser window? So it'll ask you which one, and then you select it, and then you'll be ready to go.
Right. Sorry about that.
Yeah, so are you doing it off PowerPoint or what's your?
I'm on PowerPoint. Yeah.
You should once you've got shared screen, you should be able to click on your.
Okay, so I've got you now. Is it Brendan? Got you. So I click on share screen.
Yep.
And then I see. And it says one.
And click on.
Come back.
You should be able to click on your presentation.
Okay. Yeah. Yeah. Unfortunately, it's not coming up. Oh, okay. Sorry. It just looked different in the picture. Yeah. No, I'm not a technical. Okay.
Right.
So then I go share. Okay. Right.
All right. Should be happening now, I think.
Okay. Sorry, I've got an old Mac. Well, it gives everyone time to get a cup of tea, I suppose, isn't it?
Yeah, but it's just we're on a bit of a clock.
Yeah, I know. Sorry, I've just been facetious.
So can you hit the full screen? Can you hit your PowerPoint now that you're in screen?
This is your screen sharing now. Is that better?
Okay, so if you can put the presentation mode, then off you go.
No, sure. Okay, great. Sorry about that. Just take a moment, I think. I hope. Okay. Right. Well, I'm Harold Bernhardt, and I am in Dunedin, New Zealand, sunny Dunedin, and I just wanted to thank the organisers for giving me the opportunity to talk to you today. My talk's about a proposed mechanism for peptide synthesis occurring in water and in the interlayer of clays. So the outline of my talk is I'm first going to discuss very briefly why I believe the advent of peptide synthesis was key to the evolution of life on Earth, why I believe that this origin of life is unlikely to have occurred on the Earth's surface or in a primordial soup, and finally why I believe that the answer might lie in the aqueous interlayers of clays. Okay, so we don't really know, but it is proposed that the origin of life on the Earth may have occurred as early as 4 billion years ago or earlier. And peptide synthesis or the advent of peptide synthesis would be, I think, would be widely accepted to be a critical step in this process, in the sense that all biological systems are hugely reliant on proteins for all their mechanisms, all their biochemistry. However, the problem we have is that modern biological protein synthesis is hugely complex, occurring on the ribosome, which is a complex of large RNAs and huge numbers of proteins. I also believe, such a complex system must have had a simpler origin. So that's kind of a critical point. And I also believe that understanding how peptides were first synthesized may provide insights into the evolution of other aspects of, or the origin of other aspects that were critical to life, such as RNA. I think it's unlikely that the origin of life occurred on this Earth's surface, just because of the absence in the early Earth's absence of an ozone layer, and so potential exposure to deleterious UV radiation. Also, it's not completely known when the Earth's magnetic field was established. And so prior to that, we also have the problem or potential problem of bombardment by cosmic rays and cosmic radiation. So one of the things that people have come up with is in terms of protection against we had a talk earlier, which was interesting, about the potential protection of early molecules by an oily layer on the surface of water. However, we have a problem with evolution, or early steps of evolution, of chemical evolution occurring in water. We have two problems, well, at least two problems. One is that there's a huge problem of dilution, and that's why I guess the whole idea of a primordial soup, which was once so much in favour, has really fallen out of favour, because there's just potential of almost infinite dilution of your original reacting molecules. The other problem is, in terms of the origin of peptides and RNA and DNA, is that those reactions which form those polymers are condensation reactions. So the reaction results in the production of a water molecule. And thermodynamically, that's unfavourable, because you're actually producing water in an aqueous environment full of water. And so that the actual presence of water tends to drive the reaction the opposite way you want it to go. So I'm suggesting, or I have hypothesized, and I'm not the first one by any means, to have raised the possibility of clays as the potential site for the origin of life on the Earth. And it's been proposed that clays formed early in Earth's history. For example, Mike Russell has suggested that the Hadean Earth ocean crust may have been covered in a layered double hydroxide called green rust, which is a mixed hydroxide of Fe3 plus and Fe2 plus. More recently, well, actually not more recently, but also there's been the suggestion that of marine sediment clays as a possible origin location for the origin of life. And that was by Francis Westall and colleagues, and actually Kathy Campbell, who is participating in this conference, also was a co-author on that paper. So basically, if we look at the picture up the top here on the right, that's a schema diagram. And you've got the layers, the sheets on either side, And the sheets are often charged. And because they're charged, in the case of double hydroxides, they actually contain a net positive charge. These clays, basically, they intercalate water molecules in their interlayer, but they also intercalate ions. And in the case of double hydroxides, they intercalate anions, which is very interesting for a number of biological reactions. And so I believe that within these interlayers, which, by the way, can actually swell, and that's one of the other things about clays, is that clays are basically, obviously, the reaction formed by the reaction of minerals with water, is that these clays, the interlayer can swell from about 0.7 of a nanometer to up to 40 or 50 nanometers. They can also intercalate quite large molecules. For example, recent work has looked at the intercalation, or has actually found the intercalation of large molecules of RNA within these clay interlayers. So the following reaction I have proposed is the reaction I'm proposing for the origin of peptide synthesis. And I haven't shown the whole reaction here, but basically this molecule here, which I've labeled as gamma-GOR, is formed by the reaction of ribose with a molecule of glycine, or it could be any other amino acid. And this is catalyzed in water by a small amount of catalytic amount of acid, and forms this oxazolidine 5-one, which is pictured here. And the production of this, or the formation of this ring here, actually what it serves to do is activates the amino acid for reaction with another amino acid, which I've shown as a glycine here on the right at the top. And these two react to form this extended structure. When the ribose recyclizes at the end, which is shown by this, the attack of the lone pair on this double bond, you actually get formation of a diglycine peptide, or a dipeptide of another description, with the recycling with the production of ribose to re-enter the reaction at the beginning. And this reaction occurs, like I said, in aqueous solution. I propose that this reaction will occur in aqueous solution. And what reasons do I propose this? Well, what grounds do I have for suggesting this? It's known that the reaction between amino acids and aldehydes to form peptides, and that is known from synthetic organic chemistry, a very old paper, Hiskey and Jung from 1963. And also more recent research by Laurie Barge and Mike Russell again has actually shown that green rust can catalyze a reaction which is analogous to peptide formation, but actually is one step earlier in the process, and it's the formation of an amino acid. But it's a similar reaction because it's between an aldehyde, and rather than an amine, it's between ammonia. And that occurs there's a reductive step in that process as well, and that's believed to be catalyzed by the Fe3+, Fe2+ redox couple of green rust. So I'm proposing to carry out this reaction, or to try to attempt this reaction in the laboratory, and using lab synthesized layered double hydroxides, such as hydrotalcite and green rust, and then analyze the products with mass spec and IR spectroscopy. And so I think, just in conclusion, I think I've probably shown that I believe peptide synthesis was key in the evolution of life, how it's unlikely to have occurred on the Earth's surface or in primordial soup, and that the answer may actually lie in aqueous clay interlayers. And I'm happy to take questions.
Thank you, Harold. It was a really good talk. Thanks very much. We managed to work out the technology in the end, so all good. I'm just trying to see if we have any specific questions in the chat. At the moment, we might, just because of time, we might move on, but you might, if there's questions that come up in the chat, if anyone please wants to hit Harold up, we can continue on from it there. So I might move on to the final talk before our vital talk. So thanks, Harold, and I'll just move on to the last talk, which is Tony Jia, or Jia, I hope I got the pronunciation okay, from ELSI in Japan. So Tony, if you want to, so Harold, if you can unshare your screen, and then Tony will jump in to share his.
Great, thanks. I just want to make sure, is it showing the presenter view or the full slide?
At the moment, I've still got Harold, unless any, Harold, can you unshare your screen if you? Sorry, I'm trying to. Okay, here we go. Yep. So can you share the screen now? Okay, Tony.
Yeah, let me try that again.
Excellent. Yep. So if you want to go to slideshow view, that'll. So you're in presenter view now. Is that how you want it?
Great.
Yep. Perfect. All right. I'll leave you to it. Thanks, mate.
Okay. Great. First of all, I'd like to thank the organizers of the meeting for giving me an opportunity to speak about some of the research I've been doing at ELSI and as a part of Blue Marble Space Institute of Science. My name is Tony Jia. I'm a researcher at mainly based at ELSI in Tokyo, and I'll be talking a little bit about phase-separated and membraneless compartments at the origins of life. So for those who may not be familiar, I think everyone is familiar, but just for argument's sake, life is essentially composed of cells. It's a basic unit of life, and there are many structures and functions within the cell that work together to help organisms live and survive and grow. And so one of the major unanswered questions in biological science is how the cell emerged and evolved from whatever it was before into the form that it is now. And one of the ideas is that before the emergence and the existence of the modern cell, that a primitive cell, a primitive compartment, some people call this a protocell, a precursor to a modern cell existed sometime on early Earth, and perhaps this protocell or primitive compartment consisted of some type of boundary with encapsulated genetic or metabolic molecules such as nucleic acids or peptides. Maybe the boundary is some type of lipid bilayer. So this is one of the ideas, a primitive type of minimalist cell. So then the next question is, on early Earth, how did such a primitive cell emerge? We have to understand how the chemistries that are generated from the environment and geologies on early Earth, how these simple prebiotic chemistries could have eventually resulted in the assembly of such a primitive cell, and how then we can try to understand how this primitive cell could have led to the emergence of a modern cell. And one of the things that we have to be aware of is why exactly is this cell, a compartment-like structure, crucial for life? And on the prebiotic Earth, there's many different reasons. For example, such compartments, they can concentrate and segregate reactants. They can form barriers and protect molecules within from degradation. Sometimes they can prevent diffusion of genetic materials out into the open space where they're unuseful. And maybe many of us think about a primitive cell as a structure which is containing a boundary made up of lipid bilayers. And I'd like to present an alternative type of compartment system. And so this compartment system is generated from liquid-liquid phase separation. So phase separation is a process which results in two separate liquid phases, and these form droplets. They're compartments, they're membrane-free, they don't have membranes, and they allow some exchange of molecules between the compartments themselves and the surroundings. And sometimes they can be formed from simple molecules. In fact, phase separation, these membraneless compartments have been used in engineering practices in, for example, drug delivery or chemical microreactors, and also in biology. In our biologies, they're used. So there are instances where membraneless organelles in cells, both eukaryotic and prokaryotic, so that these membraneless organelles exist and perform very specific functions such as protein expression regulation. And so some of these examples are, phage, nuage granules, stress granules, even chromatin has been proposed to be a membraneless organelle in some circumstances. And so what we wanted to know is if this type of liquid-liquid phase separation could form simple droplets that could perform some type of relevant function in the context of a primitive compartment or protocell. And so one of the types of phase separated compartments that we studied are called coacervates. coacervates are formed generally when two oppositely charged polymers, sometimes, for example, a positively charged peptide and a negatively charged nucleic acid. When these oppositely charged polymers interact, they'll phase separate and they'll form a droplet phase, which is heavily concentrated in both of those components. And these coacervates, these membraneless coacervates, could perform some very interesting and relevant primitive functions. For example, some coacervates have been shown to concentrate primitive biomolecules like nucleotides or RNA. Coacervates have been shown to sometimes be able to grow and divide based on the external application of forces such as electric fields. And there are also ways for coacervates to generate more additional complexity, such as designing of multi-phase compartments within coacervates, which each have different functions or affinities for molecules. And finally, coacervates have also been shown to be able to assemble lipid membranes around their original compartment, suggesting maybe this is some type of intermediate step between a membrane-bound compartment, more similar to a modern cell, compared to a membraneless compartment that might have existed on early Earth. And so what we wanted to do in this study is to try to find another way by which a coacervate could increase its structural complexity as a way to perhaps find other relevant structures or functions that could have been present on early Earth. And one of the systems that we decided to use is through DNA liquid crystals. So DNA liquid crystals, they form when nucleic acid oligomers, they'll form duplexes, and depending on the length, they'll form these rod-like structures through end-to-end stacking of these small DNA duplex cylinders. Eventually, when they become rigid enough, when they become long enough, they form a liquid crystalline phase, which is liquid in character, but has some interesting properties, such as it can modify the surface chemistries, change stereochemistries of reactions, change electrochemistries. So we wanted to see if we could generate or find some type of system where these DNA liquid crystals could co-assemble with a coacervate to see if we could make a kind of a liquid crystal coacervate by incorporating a cationic polymer system, a cationic peptide system, polylysine, with these DNA liquid crystal duplexes. And so at first, we started to try to find conditions which allowed this interaction to occur. And we were able to find that a DNA 12-mer duplex, self-complementary duplex, could bind with a long polylysine of around 200 bases, and it can form not only this nice membraneless droplet formation, but also a pattern through polarized microscopy indicative of co-assembly of the liquid crystal. And then we next tried to see whether what types of structures could be achieved when the salinity was changed. And so in this case, we actually ended up using a salinity, a salt concentration of about 800 millimolar sodium chloride. And we were able to see that modulating the sodium chloride concentration through changing the binding interaction strength between the peptides and the DNA, or between the DNA themselves, they ended up forming different types of structures. For example, at low sodium chlorides, they formed some type of precipitate formation, whereas at higher concentrations of sodium chloride, the liquid crystal either disassembled or the droplet itself even disassembled. So next, we then wanted to see if there are some type of relevant prebiotic processes that could also affect some types of changes in the structure of these droplets. And so here we subjected a system initially in a precipitate state to dehydration. And we saw that, through dehydration, we actually changed the system through all of these different liquid crystal phase states, from a precipitate to a co-assembled liquid crystal coacervate, to a non-liquid crystal coacervate, to a uniform phase. And this is likely due to the fact that dehydration at the same time increases the salt concentration that's dissolved in the system. And then finally, another environmental process that we wanted to test was heat. And so we want to see the effects of heat on the structures of the liquid crystal coacervate system. So again, starting from we believe to be somewhat inactive precipitate state, by increasing the temperature to around 60 degrees, the precipitate state actually transitions into a colocalized liquid crystal coacervate state. And then up to 70 degrees, the liquid crystal formation actually dissociates, probably because the end-to-end stacking of the DNA duplexes starts to get disrupted. And then of course, on the way down, we saw eventually the reformation of these colocalized liquid crystal coacervate states. But upon decreasing temperature, we actually found different states of liquid crystals. So these liquid crystal states are columnar phases. They're very different from the cholesteric phases that we saw before. And so we believe that doing this annealing type process actually subjects the system to eventually reach its most likely most stable conformation, which is these columnar phase liquid crystals, where liquid crystal droplets, whereas previously it actually started at a precipitate state. And so undergoing the annealing process at different salinities, we actually saw, of course, again, that different types of liquid crystal phases, different mesophases could form despite starting at an original precipitate state, for example. And so we believe that some of these states, the amount of packing and the hexagonal packing of the DNA is actually one of the things that's governing what types of states that we see in these structures. And so, of course, we actually in this study used a pre-designed DNA duplex of a 12-mer. It has been shown that liquid crystals can form from shorter oligomers of DNA, like 4-mers, or even in some cases, nucleotides. And so one of the next steps that we want to do is to minimize the system so that we can try more prebiotically relevant structures, such as single nucleotides, dinucleotides, something that's a lot more easy to synthesize in a prebiotic environment. And so this study has just been published recently in ACS Nano. If you're interested in learning about the details, there's a lot of stuff that I didn't talk about in this presentation. Please go ahead and take a look at this paper. And there are some other systems that we've been working on that are also membraneless primitive compartment systems, such as polyester microdroplets. And so these are two other publications that if you're interested to learn more about membraneless primitive membraneless compartments, please take a look at some of these studies as well. So I'd like to thank all of my funding sources, all of the people who helped to make this work happen, especially my main collaborator on this study, Tommaso Fraccia, who's a researcher at ESPCI in Paris. And I'd like to make a short announcement about AbGradCon, which is the Astrobiology Graduate Conference. This is a conference for graduate students and postdocs in astrobiology research. It was canceled this year, but we're hoping to hold it again next year in person at ELSI in Tokyo, and the new dates will be September 14th to 17th. So please look forward to more information online through social media or through our website, abgradcon.org, sometime soon. So thank you very much, everyone, for your time and attention. Thank the organizers again for giving me this opportunity, and I'm happy to take any questions right now.
Thank you, Tony, and good to get a plug in there for AbGradCon, and hopefully the pandemic will allow us to revisit this in a year's time. So any questions? I'm just, if anyone has any specific questions for Tony. Sorry, I'm just kind of wandering through the chat. Got one from Anna down here. What happens if you have random other DNA in there?
Yeah, that's a good question. We started to try doing some studies with random DNA, not in combination yet, but just by themselves. And I think it was important for us to show the system first so we knew a little bit more about what kind of conditions were required for the co-assembly. And it will be interesting to see what other random DNA can do in the system and, you know, mixtures as well. One of the things that we did do that I didn't show was it's actually very easy to disassemble the, or prevent the co-assembly of the liquid crystal in the coacervate phase by simply designing the DNA duplex to have sticking ends out. So if you design the DNA duplex to have two base a 2-base overhang on both sides, those DNA duplexes can't stack upon each other, and it's impossible to form a liquid crystal phase. So it'd be interesting to learn a little bit more about how this type of activity is affected in a coacervate phase.
Excellent. All right, thanks, Tony. Actually, it's perfect timing, so we might move on now to our invited speaker in this session. So thanks, Tony. You can unshare your screen, and then great pleasure to introduce Dr. Sudha Rajamani. If you want to share your screen, Sudha. So,
hi, Brendan. It says the host has disabled. I can now. I can. Yeah.
So
sorry about that.
I've just got your little intro here, which the one bit that interests me that says an unplanned set of events set Dr. Rajamani onto a current astrobiology, which sounds intriguing to me. So I don't know if you're going to touch on that, but really briefly. So Sudha is now heading there.
Oh goodness.
We appeared to have lost Brendan there again. He's having some issues. So my name's Erica. I'll just continue what I think you might have been saying to welcome Sudha today. Just a little bit about Sudha and her bio. She received her PhD in biochemistry from the National Institute of Immunology in New Delhi in India. Her thesis was about delineating the biochemical properties of sickle cell hemoglobin in a specific context. She then joined the Fink Lab at the University of California at Santa Cruz as a postdoc fellow. And then this is what Brendan was talking about. An unplanned set of events led to her current field of astrobiology. So we'd welcome you to share what you mean about that, Sudha. She's currently heading the Chemical Origins of Life Lab, also known as the COOL Lab, at the Indian Institute of Science, Education and Research. Dr. Rajamani and her team are working to help discern the series of events that might have led to the origin and evolution of early life on prebiotic Earth. They use tools from diverse areas to delineate relevant processes in the aforesaid regard. So having said all that, Sudha, thank you very much. I can see your presentation's up, so I'll let you take it away. Thank you.
Thank you. Can you hear me?
Yes, everything's great.
Awesome. Very good morning to everyone. It's about 9:30 in India now, and I'm not a very morning person. So the last couple of days to wake up at 5:30 to plug in was a bit of a challenge, but I've loved every bit of it. And thanks a ton to the organizers for putting this amazing meeting together. And it's an honor to have been invited to give a talk. Particularly a shout out to those talks that bridged science and art and talked about how to use science outreach as a way to reach indigenous and minority communities. I just thoroughly enjoyed those talks. As for me, I'm Sudha. I'm currently heading the COOL Lab at IISER Pune. The COOL stands for Chemical Origins of Life, but actually the truth is we are also really cool folks. So yeah, anytime you want to talk to us, just holler or check us out and then we can take it from there. I'm predominantly going to be giving you an overview of the prebiotic processes that we have been looking at, which we think have important consequences for the emergence of protocells. And particularly, I'm going to be talking about how selection pressures that would have been present on the prebiotic earth would have shaped these processes. Chaitanya Mungi is the first student who joined me, and soon after that Nirja joined me, and they kind of helped get the lab up and running. And the two of them looked at two different processes. Chaitanya predominantly focused on looking at origin of informational molecules, and Nirja was very interested in understanding how, once these informational molecules came about, how might they have replicated that information faithfully, even in the absence of elaborate and fantastic machineries like the proteins. More recently, in the last 4 years, Manesh, Sushovan, and Shika joined me to take this work further on. Manesh has already talked to you about the work that he's been doing. Very exciting stuff, looking at protocells and how they respond to their environmental conditions. Sushovan is also extremely interested in looking at what might this landscape have looked like on the early Earth and how would the selection pressures have shaped them. Shika's taking on a little bit of what Nirja did, and she's also interested in other questions like primitive metabolism. But for today, I'm going to focus on origin and evolution of informational molecules and the emergence of protocells on the early Earth. So trying to move to the next slide. Yeah, there we go. So this is just to give you a sense of the timeline that we are looking at. And we're looking at processes that might have occurred about 3.8 to 4.2 billion years ago. There's enough reason to believe that very interesting prebiotic chemistry set the stage, and we had all these wonderful monomers and precursors of monomers that are relevant to extant biology. And from then on, a lot of interesting processes occurred over billions of years to eventually lead us to this time in the early history of life on Earth, which is referred to as the RNA world. There are a few schools of thought in the field, but RNA world seems like one sort of theory that everybody agrees upon. How it came about is the big discrepancy or the big debate that keeps happening. But it's suffice to say that RNA molecule probably was the main biomolecule that was used by very early life forms because it can both encode information and catalyze reactions. But it's increasingly becoming very clear to us that prior to this RNA world, we might have had other pre-RNA worlds where more simpler informational molecules might have emerged much more readily from the prebiotic soup. And I'm going to talk about these processes with which essentially allowed for the transition from non-life to life on the early Earth, with a very crucial contribution coming from lipids. Why? Because it particularly sets the stage for the emergence of cellular life on the early Earth. So these are broadly the steps that we think would have led to the emergence of primitive cellular life on early Earth. A lot of prebiotic chemistry leading to building blocks, non-enzymatic oligomerization processes that would have allowed these building blocks to form much more interesting oligomers, which went on to evolve to become biopolymers. And in this space where you might have had a lot of oligomeric molecules of RNA present in the RNA world, once in very interesting catalysis of functions come about, one needs to think about how might they have faithfully replicated this information, or to what extent would they have faithfully been able to replicate this information in the absence of fantastic machineries called proteins, because this whole nucleic acid protein business leads to this chicken and egg problem. What came first? And RNA world in that sense provides a respite. And simultaneously, during all of these processes, you would have had the formation of simple to complex protocellular entities, and there would have been a constant crosstalk between biomolecules and biopolymers and lipidic entities, amphiphilic entities, to eventually lead to the emergence of protocells. So what are the environments that would have supported these kinds of reactions, right? Where you could go from monomers to oligomers, where you can assemble protocells. That's been the focus of the first half of the day, so I'm not going to dwell too much upon it, but just to say that there are two schools of thought again. Some say that it would have predominantly been facilitated in the hydrothermal vents present on the seafloors, whether they be it alkaline or acidic, and different vents would have supported different kinds of chemistries. Another school of thought, which sort of I subscribe to because it's much more tangible for me to think about these processes in these environments, talk about terrestrial geothermal pools, talk about hot spring systems where these kinds of reactions would have been facilitated. And here are pictures of some of these niches from different parts of the world. Now, the good thing is that in the last couple of decades, enough work in the realm of prebiotic chemistry has shown that a recipe for life itself would have been plausible given the right ingredients, right fundamental ingredients, right? They have shown that you can go from these ingredients to amino acid precursors, to nucleoside and nucleotide precursors, and to lipid precursors. So one can then say with some conviction that yes, you had all of these present, so that so it's important to try and understand how you go from these precursor molecules to a protocellular entity. There's been a lot of emphasis on this today. We very recently sort of got the courage to step out of the lab and to try and understand these processes in realistic analog conditions. What do I mean by that? I mean, a lot of the experiments that we have done this far from 1953 onwards, when the stage was set with the Urey-Miller experiment, has predominantly involved us working in the confines of the lab, trying to simulate a lot of these conditions in the lab. And when we try and do these experiments, it becomes extremely complex very quick if you're trying to parameterize it with too many different conditions. So what then people tend to do is to try and understand the processes at the fundamental level. You sort of scale it down big time. Yes, it allows us to understand a lot of the mechanisms that are involved in these experiments, but you lose out on the context, and the context plays an extremely crucial role in how these might have actually panned out under realistic settings. And I'm sorry, I don't have extremely blingy looking pictures like Manish did, but these are the 3 hot springs from which we brought back the water samples, and we have used this quite a bit to try and understand the 2 important processes that I've been talking about thus far, which is emergence of informational molecules and formation of protocells and their sustenance under these conditions. So we work with this model in mind, which is essentially saying that under prebiotically plausible wet-dry cycles, which is even prevalent on modern Earth, one could facilitate the emergence of protocells if you had the right monomeric entities. So here in panel A, you're seeing this is sort of amphiphilic monomers which exist at the edge of air-water interface. They can form these higher order structures like micelles, bilayers, and vesicles. And these red little small rods are supposed to be the monomers of informational molecules. And in a 3-dimensional space, what happens is these molecules cannot really interact with each other very efficiently, but during a dehydration stage, which would have been possible at high temperatures of the daytime, you can have these molecules be constrained in a 2-dimensional space where now you can have much more efficient interactions between them, resulting in these small chain-like entities, essentially a string of pearls, which are informational molecules. And on a subsequent rehydration stage, you can have them bleb out with the vesicular, with the lipidic entities to form these vesicles, which are very reminiscent of protocells. And Bruce has already touched upon it. Progenote-like communities could have probably existed where a lot of interesting evolutionary processes would have kicked in, even which were simply driven by the physicochemical parameters of the early Earth. So first, to talk about informational polymers. Formation of polymers, extremely crucial step in life. Everything is driven by polymers. Your information coding, information processing, facilitating functions, all polymeric in nature in extant biology. So it's crucial to understand how they emerged. Polymerization in modern biology is ultra complex. Again, the chicken and egg problem. So how can we think about it? We invoke the RNA world hypothesis, where we first try and understand how did the nucleic acid entities come about, which then the oligomeric nucleic acids could evolve functions, and they're called ribozymes, which would have then kick-started the stage for further evolution of life on Earth. So early polymerization had to be non-enzymatic. No enzymes, no sexy machinery around, right? So then what would have happened? You could essentially envisage that monomers come in close proximity, and under low water condition, water activity or lack of water during the dehydration stage, they can undergo what is called a condensation reaction, where you can kick out a water molecule and form the very crucial bond that links these monomers together. This is good for amino acids, which lose a water molecule, come together to form a peptide bond. This is good for nucleotides, which lose a water molecule, come together to form a phosphodiester bond, so on and so forth, yeah? Now, there's been a lot of work in the field to begin with, where polymerization was essentially understood using activated nucleotides. What do I mean by activated? They are chemically modified in some way to be able to follow through these reactions on realistic timescales, and more importantly, be able to understand the product distribution, because they just result in wonderful chemistry. However, that's not very realistic. So ever since I worked with Dave for my first postdoc in this field, and I stumbled upon it by chance, and I'll get to it later, there's been a big focus on trying to understand how you go from non-activated nucleotides to polymers under realistic conditions of the early Earth. It's very difficult to work with them, extremely difficult. At some level, you might think that we are sort of kicking ourselves in our own foot, but hey, life started at some point, and it happened over billions of years, and it's very plausible to systematically figure this out. So what do? We simulate fluctuating environments that would have been present on the early Earth, and very interestingly, these fluctuating environments are super cool because they can facilitate uphill reactions like oligomerization. And if you had lipids in the vicinity, some of the back reactions, which are very readily possible when you have a rehydration cycle like hydrolysis, degradation of the oligomers and monomers can potentially be prevented because they can start encapsulating these growing oligomers within these compartments, protecting them from the harsher outer conditions. Here is an image which we kind of shamelessly mooched off of Bruce and Dave's work, and basically it shows some of these regimes in which these reactions would have been possible, where high temperatures, varying pH, dehydration, rehydration cycles are facilitated, and the environment in itself can drive kinetic traps that can now protect these oligomers and prevent them from going back to their monomeric stage. Yeah. So we focused a lot on lipid-assisted synthesis of oligomers. It started with the work that I did in Dave's lab, where we showed that you can form RNA-like oligomers when you do this wet-dry cycling of the RNA monomers in the presence of lipids. And it seemed like we could form oligomers which were 100 nucleotides long. And this is already something I talked about. In the you can constrain these molecules, monomers, and then facilitate some of these cool reactions. And more recently, Dave's group has shown that double-stranded RNA is relatively resistant to hydrolysis. So it still sort of gives us the confidence that we can try to understand how these molecules came about. So we started out with wanting to characterize these reactions systematically because, you know, I say RNA-like oligomers, and we wanted to understand what exactly we meant by that. So what we did was we tried to understand various parameters on this process, like temperature, pH, drying time, presence of lipids, the kinds of monomers we use. Why is this important? These would have been the very crucial physicochemical parameters that would have played the role of selection pressures on early Earth, and it is extremely crucial for one to systematically try and vary them to understand these processes first within the constraints of the lab, and later on go out into the field and try and understand these processes by retrieving samples from different kinds of hot springs which have different pHs, which are at different temperatures, so on and so forth. So we also tried to standardize conditions for maximizing the yields from this reaction, and we went a step ahead and we tried to make our lives worse by trying to put all the nucleotides together at one go. So how does the setup look? Nothing like your volcanic geothermal pool. It is a simple heating block, but it allows you to really do a lot of things which essentially would happen in a volcanic terrestrial geothermal volcanic environment. These are the representative monomers that we worked with. Here is your adenosine 5' monophosphate, not activated in the sense that your phosphate doesn't have another chemical entity stuck on it, which allows it to be much more reactive, so it's non-activated. And here is a sort of a go-to representative amphiphilic entity that a lot of people in the field have used to sort of make the protocellular membrane structures, which they're a sort of a proxy for prebiotic amphiphiles. And we do these wet-dry cycles by bringing them together, and we do all of these under anoxic conditions to under carbon dioxide to simulate some of the early Earth conditions. And how do we analyze these products? We use a lot of different different bioanalytical techniques. HPLC, which is high performance liquid chromatography, is one of the way to look at it. So over multiple cycles, other than this monomeric peak, you start getting dimer, trimer, tetramer, so on and so forth. But as you can see, beyond tetramer, it becomes extremely difficult to sort the products out. So we then go to an even more sensitive technique, which is called mass spectrometry. And this is a representative image of the instrument that we use, an LCMS instrument, which allows us to systematically characterize the products that have been formed in these reactions. And then you may ask, okay, Sudha, you're subjecting all of these reactants to very harsh conditions, 90 degrees C, acidic conditions, or alkaline conditions. Would your membranes survive? You'll be surprised to know that the membranes are extremely sturdy, and we follow their stability. There is an indirect readout of their stability by trying to do microscopy, which tells us if they retain their character to form vesicles even after multiple cycles. I'm showing you only till 7, but trust me when I say we've gone up to cycle 25 and they're fine. So what did we do? We varied pH, and what did we find? We found that the process is most effective at pH 2. You see, more number of peaks. Of course, there's breakdown, but if you go above pH 2 to 3 and 4, you start having much less oligomerization, but then there's also much less breakdown, which is great, but oligomerization doesn't happen very effectively. Go down to much lesser pH. It's not very evident from this, but everything kind of goes kaput. It all breaks down. And we even weighted the different kinds of acids to sort of understand how the ions in the system, ions present in the environment, might affect this process. And of course, there is an effect that one can see. As you can see, H2SO4 did better than the rest. Again, temperature has a very important role to play. Darwin's warm little pond, which is closer to 60 degrees C, is fantastic, but it doesn't facilitate a lot of these reactions because you need to pull water out, and it takes a very long time to pull water out at 60 degrees C. So if you start increasing the temperature, it gets better and better, but then there is a trade-off between your breakdown and your oligomerization. So one sort of tries to stick between 75 to 90 degrees C to understand these processes. And your wet-dry cycle drying time also seems to play a role, though it is not as prominent an effect as is the case with the other situations. And we tried this with other nucleotides. It just doesn't work with adenosine-containing monomers. It works with GMP, with uracil-containing monomers, with cytosine-containing monomers. And then, like I said, we make our lives hell all the time. We mixed all of them together, and it still works. However, the important caveat is that the product distribution is pretty messy. What do I mean by that? We set out to form informational polymers, but what we noticed is we've ended up forming what is called abasic polymers. What are abasic polymers? If you take just this entity here, this is a dimer of a AMP molecule. You see that the informational moiety, adenine, is present only on one of the ribose while it's absent on the other. This was something that we kept seeing again and again and again. So this made us wonder. This is one of the important crossroads where we took a detour, and Chaitanya and I wondered, you know, AUGCT are all bases that have been selected for over billions of years, but maybe early Earth had other kinds of nucleobases which were much more sturdier and could survive the harsh conditions of the early Earth. So then the search for alternate heterocycles started. We are not the trailblazers at all. This was done all the way back in 1971 by Leslie Orgel. Any prebiotic chemistry research that you want to do, I will say, please go and look for Leslie Orgel's papers. There might be something out there that Leslie and his team has already tried. And more recently, our inspiration came from the HUD group, where they systematically tried to do this pen and paper thing, where they put together different kinds of heterocycles, 6-membered and 9-membered rings, to see what might be plausible, what is seen in extant biology, what has already been shown in prebiotic reactions, so on and so forth. And there's also been work from the HUD lab where you see here this 2-pyrimidinone. It looks so much like your uracil and cytosine. Only thing is some of the hexocyclic groups are missing, but it can very readily assemble to form the nucleoside that's shown here called zebulorin. And there's work even from Benner and group where they showed that 6-amino-uracil, which is just one extra amino group on the uracil molecule, can very readily form nucleosides under the harsher conditions which I'm talking about. Now, what we ended up doing is we thought of looking at certain other heterocycles that hadn't been looked at before. We predominantly work with barbituric acid, but we have tried working with these other entities shown here. And it's very intriguing that unlike modern nucleobases, these nucleobases very readily can assemble to form these higher order structures, which allows us to think about alternate ways of how informational molecules might have come about on the early Earth. But essentially, what did we do? We took a ribose monophosphate. We took one of these alternate bases. We put them through wet dry cycles or dry heating to show that you can very readily form these nucleotides, which otherwise is not plausible with some of the modern bases. And these are just HPLC profiles showing that we indeed can go from the starting material to the product. And it's shown in these HPLC profiles. The cool thing is that this nucleotide also pretty readily oligomerized under harsh conditions, right? So this is just to give you a sense of how you can go from the monomer to, this is the breakdown product, how you can go from the monomer to a lot of these other oligomers. And you can see that there's a lot of different isomers that can be formed, because you can form both a C glycoside linkage and an N glycoside linkage, depending upon the atom that, depending upon the atoms that are involved in the bond formation, glycoside bond formation between ribose and the nucleobase. And what I was talking to maybe a slide or two ago is that these higher ordered structures that are readily formed by these prebiotic nucleobases, so to speak, and cannot be readily formed by extant bases at all, allows us to think about this alternate possibility of how informational molecules would have formed, which is via this polymer fusion model. The recognition units can very readily come together and form higher ordered structures, either these diodes or these much bigger assemblies, hexad assemblies. And the backbone could have later on sort of assembled around these entities. And this is something that has been extremely heavily propounded by Hudden Group. And we also went on to show that indeed you can do a polymer fusion kind of thing with the BA nucleotide. And around the same time, we, both HUD and HUD group, and we published this work where we showed the formation of this prebiotic nucleotide, so to speak. So then this made us think, you know, if we can do this polymer fusion kind of thing, can we just readily form the backbone? Forget about the informational moiety, but higher ordered structures that these informational moieties form. So then we started thinking about, can you hear me? It says my internet connection is unstable. Hello?
Yeah, we can hear you okay. Keep
Okay,
going.
awesome. Sorry about that. So then we started off just with the ribose monophosphate. We said, we don't care about the informational moiety. Let's take it out of the picture. And we wanted to see if wet-dry cycles under early earth conditions would facilitate formation of these kinds of backbone entities, right? That's highlighted here in this yellow sort of a rectangle, because very early on, prior to the emergence of RNA DNA in this pre-RNA world, you could have had any kind of backbone, which should have an ionizable sort of an entity at the back, and then you could have had any kind of trifunctional connector instead of the ribose, or any kind of recognition unit. But we sort of thought we'll start simple first and try and see if we can even oligomerize the ribose monophosphate to form a backbone. These are called sugar phosphate backbones, and they very readily could form, and we systematically showed with mass analysis that you can indeed form these backbones. And we are now at this stage where we are trying to see if we can systematically demonstrate polymer fusion hypothesis in a more rigorous manner, where we start with known starting reactants and can show the mechanisms by which you can form these products in a very systematic way. So when we realized that these 5' monophosphates were leading to, use of 5' monophosphates was leading to these abasic oligomer formation, we figured, you know, we can try using one other monomer, which is non-activated in the chemical sort of way, but it is intrinsically more activated than the 5' monophosphate. And these are cyclic nucleotides. And this is work that was, that's been done by Shikha, and we recently published this in RNA, where she showed that you can use both 2,3 cyclic nucleotides and 3,5 cyclic nucleotides to essentially form oligomers. And she extensively used mass spectrometry to show this, and here is just showing you one representative of the mass spectrometric analysis, where you're seeing the AMP dimer, and she went on to show that AMP can readily form trimers and tetramers, intact informational oligomers, and your CMP moieties can form intact dimers and trimers readily under these conditions. And to point out, she also did one other thing. She sort of did these realist, pure lab simulated reactions, so to speak, and she also challenged herself by doing these same reactions using one of the hot spring samples, and she showed that something interesting is happening there. We are still trying to understand what is happening there. But having said that, it is possible, there is a hypothesis that under these realistic conditions, maybe we would have selected for what we are seeing, the phosphodiester linkage that we're seeing in canonical nucleic acids, which is the 3,5 phosphodiester linkage. Because as I showed in the previous slide, if you start with a 2,3 cyclic nucleotide, you can either form a 2,5 linked dimer, or you can form a 3,5 linked dimer. It's essentially the position of the sugars where the linkages are formed, right? So 2,5 is not seen in extant biology, but 3,5 is what is seen. And her work with the hot spring samples or water samples is showing that maybe just realistic conditions in realistic analog conditions, the ions that are present there might have itself helped in this kind of selection process. Neerja worked extensively on trying to understand non-enzymatic replication and how that might actually pan out under a heterogeneous prebiotic condition. I mean, typically you use template primer nucleotide, boom, do the reaction in test tubes, wonderful results. What if you now challenge yourself and try to simulate some of the heterogeneity that is inherent to the prebiotic zoo? That is what she did. And she went on to study extensively this one particular paradigm, wherein you have a template nucleotide, nucleic acid, you have a primer short primer oligomer, and then you have these incoming nucleotides, which then allow and you kind of can facilitate these reactions under certain conditions, and you can nicely then form a complementary strand which uses the information in this strand to make a new polymer. So what she went on to show, I'm just kind of encapsulating in one slide, she showed that in the presence of co-solutes, you can have higher mutation rates. Immediate thought would be, oh shit, higher mutation rate, not good. But turns out higher mutation rates are not bad at all. Higher mutation rates would have possibly allowed for exploration of a larger sequence space much more readily, which is very crucial for the evolution of ribozymes in an RNA world. And this is a piece of work that we have started to collaborate on with Julian from University of Paris Diderot and Supratim from IISER Kolkata. And we just recently have gotten a paper out on Journal of Theoretical Biology where we show that if you can use these realistic polymerization rates and simulate more realistic early Earth conditions, one can actually see tRNA-like structures and simple clover leaf, hairpin-like structures very readily emerge from the prebiotic soup. We did this using theory and modeling with real rates. And now we are also working on using biophysical tools to understand the mechanism behind this observed effect. Why is there a higher mutation rate? It was a little unintuitive to us that all the mutation rates would go bad. We thought some of the more faster reactions would have the bigger problem, but everything was bad. And we have been trying to understand RNA-lipid interactions to try and understand if they play a role. We've realized that nucleotides can interact within themselves, and that also leads to an interesting effect. And all of this work has already been published, and you're welcome to go and check it out. Second and last, the last third part of the talk, which is where I'm going to focus on the compartment. So we've talked all about molecules of emergence of oligomers in an RNA world and their replication. That is one important prebiotic process pertinent to the emergence of protocells because it leads to the formation of informational and functional molecules. The other part is where you have the compartment, and one is forced to think about how these compartments might have emerged and survived under the harsh conditions of the early Earth. What do I mean? What are the selection pressures that they would have been subjected to? Dilution, which is something that people have talked about in previous talks. The pH, you have a whole range of pH regime, and depending on who you talk to, they would say that life emerged in this regime versus that regime. But the truth is some of the prebiotic processes are much more readily facilitated under acidic conditions, whereas certain others are much more readily facilitated under basic conditions. So it might be worth our while to systematically think about this prebiotic landscape of amphiphiles to try and understand what might be those groups of amphiphiles which can actually span this whole set of pH. And then you also have, very importantly, divalent cations. Why are they important? Divalent cations are super crucial for functioning of the ribozymes, RNA enzymes. But having said that, these divalent cations can cause trouble for these protocellular membranes if they are in vast excess. They are crucial. You need them to help with the counterion, to help with reducing the charge repulsion that comes from the head group of the membranes, but you can't have too much of it, too much, too bad. So these are the kinds of questions that Sushant started to ask. And enough work in the field has looked at fatty acids, which are thought to be prebiotically more relevant because they were very readily formed under early Earth conditions and in Fischer-Tropsch-like reactions. And there's enough proof that they would have ridden on meteorites and during the late heavy bombardment that is being heavily debated if it was really an intense period or was it spread out. Those kinds of events would have brought in a lot of these entities onto early Earth. Murchison meteorite that Dave and team have worked on showed that you actually have these hydrocarbons up to C12 chain length. So a lot of chain lengths, head groups have been varied, and here is sort of an ensemble picture of what all different kinds of head groups people have worked with. And in the past, people have shown that if you dope a surfactant with the amphiphile that you're working with, either a fatty acid or an alkyl phosphate or a cyclophospholipid, you can increase the pH regime in which these can form reactions. However, they looked only at one or the other process. What we ended up, and there's one very important piece of work where people moved from a binary system to a tertiary system with 3 components, and there Mansi showed that you can actually, compositional heterogeneity of the protocell means your vesicle formation is much more readily possible, and they can also be thermostable, stable at high temperatures. What we wanted to do is systematically try and understand this process, both mechanistically and to understand what the compositional heterogeneity, increase in compositional heterogeneity might mean for these systems. We predominantly focused on a C10 and C18 chain length system. Here I'm going to show only results from the C10 system because the argument is that you would have shorter chain length fatty acids in more abundance than longer chain length fatty acids. So it makes more sense to share this result. And what we started off is to understand what happens to the self-assembly and to some of the important properties that are determined by the selection pressures that I talked to you about. How did we look at the self-assembly? We can do it by using interesting fluorophores that allow us to understand when these monomeric entities go from being in a disordered phase to an ordered phase. And that is what is encapsulated in this graph here. And this black line here gives us an idea of the lipid concentration about which you start having ordered assemblies now, yeah? So this is called the critical vesicular concentration or the critical aggregation concentration, more broadly speaking. And if you, this slide is to encapsulate the fact that if you just had a pure fatty acid all by itself, yes, it can form vesicles, but only at a high concentration, 30, 32, 35 millimolar. But you start doping it with other derivatives of fatty acid. Here is glycerol containing 10-member fatty acid. Here is an alcohol head group containing fatty acid. I'm sorry. Sorry, these are not C10 systems. These are C11 systems. Pardon me. This is not decanoic. This is C11 systems. You can see that the more you start doping and the more compositionally heterogeneous it becomes, your critical aggregation concentration now shifts to the left, which means they can much more readily form these higher ordered structures. We also went on to systematically try and understand this with microscopy, and the take home of this panel is, see, you're looking at pHs, yeah, column wise, row wise, it's the systems, just the fatty acid system, your binary systems with glycerol and alcohol, and your tertiary system where you have the fatty acid, the glycerol head group containing entity and the alcohol head group containing entity. What this shows is that the more the complex the system, the more readily it can form these vesicles, these beautiful globular looking structures over a wider range of pH, which is not plausible when you're just working with the fatty acid. Below a certain pH, which is below the pKa of the head group, you only form oil droplets, and above that pH, you don't form anything. But then start doping it with other things, start making the system more complex, you start seeing these beautiful entities surviving at much larger pH scales. This is just to show in one shot how did these C11 and C18 systems sort of behave when you kind of sort of made the starting reactant mix more complex. Here is your fatty acid, purple, yeah, light purple and dark purple, much more narrow regime of pH is where they form vesicles. But as you increase the complexity of the system, the pH regime is like super wide. Now, we also wanted to understand what happens to cations, what the role of, so if you have too much cation, like I told you, you will destabilize these systems. So we wanted to see if compositional heterogeneity of the protocell might somehow rescue this problem. And indeed, we did see that. Here is just your fatty acid system. By the time you hit 4 millimolar of magnesium, you start forming crystals. Your vesicles are gone. I mean, you might see a couple of them, black arrows. The red arrows are disordered structures, essentially crystals in this case, but you dope them with a second derivative, a second amphiphile, and then you start forming these vesicles at a much wider range of pH. And depending upon the derivative you dope it with, you can have different effects, and I can talk about it if anybody has questions. But then your tertiary system is the coolest, right? You can go up to 16 millimolar magnesium or 24 millimolar magnesium, and they very readily form vesicles. Can we rescue this effect? Are these crystals forming only because of magnesium aggregation? No, they're forming because of magnesium-induced aggregation of vesicles, and we can rescue the effect if you actually add a chelator like EDTA, which can chelate magnesium, and then you can form these vesicles very readily again. Now, we also did leakage assays to try and understand the dynamicity of the membranes that are resulted from either the homogeneous system or the binary system or the tertiary system. Suffice to say that the more complex the system becomes, the more interesting it becomes. You see the UDA GMO system, which is essentially just the fatty acid, and the binary system containing the glycerol monoester head group. So here you're seeing a fluorescent entity encapsulated into the vesicle, and we look at how this leaches out over time. And this is calcine. So you start with about 100% of calcine in the vesicles, and over time it leaches out. Why is this important? You did not have proteins in the membrane to pump things across the membrane to get the important stuff in, to get rid of the unnecessary stuff. You didn't have these heavy duty protein machineries in these membranes. So the membranes had to be intrinsically dynamic to be able to undergo these processes. And we tried to understand what compositional heterogeneity meant for this process. And finally, we did one thing which nobody else has done before, which is trying to understand vesicle stability when you subject them to multiple selection pressure in a sequential manner. Why did we not do it all together? Because then discerning what is affecting what becomes problematic. So we applied the selection pressures in a sequential manner. What did we do? We first diluted the system out. And even under high dilution regimes, the composite systems formed membranes more readily. After subjecting them to dilution, we kind of threw magnesium at it. Then you can see that the binary systems kind of lost out a little bit, though some of them did work fine, but the tertiary systems are the ones that had these vesicles retained for a very long time. And finally, you take the same system which has been subjected to dilution and an onslaught of magnesium ions and then subject it to alkaline pH, high alkaline pH, right? pH 11 or something, pH 10. Only the tertiary system survives. Strengthening the notion and the understanding that compositional heterogeneity would have had a huge selection advantage under earlier conditions. This is just showing you the same thing, but we first did the magnesium ion onslaught, then alkaline pH, then dilution. It doesn't matter how you apply the selection pressure. We varied it in every possible combination. Suffice to say, the composite systems work the best. So we are at a stage where we are trying to get to a point where we first try and understand how these model protocells or the progenote-like systems assembled, and how then did the transition happen from the model protocells to the more contemporary compartments via these intermediate cells. Very cool work has come out from Jack Shostak's lab and from Sahai's lab, Meeta Sahai's lab, where they've worked with these intermediate systems where you have both the fatty acids and phospholipids coming together to form these ensembles. However, the dynamicity of these systems are still a little sort of iffy, and we are trying to understand better how intrinsically they can be, how you can invoke realistic prebiotic processes and try and understand more about the dynamicity of these vesicles. So finally, what is it that we can do? The best that we can do is to draw up this story, which is consistent from all these different results that we get, all the evidence that it talks about, right? Whether it is prebiotic chemistry experiments, whether it is trying to understand the earliest life forms that were present on early Earth, or trying to understand the minimal biology or the minimal lifelike systems from a top-down approach to try and understand how these might have worked. The best we can do is to come up with the story that is consistent with all the evidence, and happy to say that after this after several decades, the story is finally coming into view. And this is a quote that I shamelessly mooched off of Irene and Andrew's paper. And finally, to thank the important players, Cool Lab members. We wouldn't have been able to do what we did without you cool folks. IISER Biology, Chemistry, IISER Pune Biology, Chemistry, and Earth and Climate Sciences. We have a lot of collaborations with colleagues in these departments, funding from both IISER and a few Indian agencies like SERB and DBT. Importantly, special thanks to LC Origins Network. I really wish it still existed. It allowed us to foster this beautiful collaboration with some of our colleagues and now friends from LC, which kind of pushed our work really far. That was really, really cool. And thanks to CNRS for funding that led to the theoretical paper work that I'm doing with Shupratim and with Julian. Thank you all for your attention. Happy to take questions. I'm sorry, I hope I didn't overshoot my time.
Thank you so much, Sudha, and apologies that I cut out halfway through introduction. My Zoom cut out. Many thanks to Erica, who's not only more than capable, but she's more than capable than me. So, really, really, really good talk. If anyone wants to jump in and actually unmute themselves, I can ask a question. I'm looking in the chat. There's one from Luke Sudha. The lipid that you're using, PLPC, how prebiotically realistic is it? And do more prebiotic fatty acids have the same result?
So sorry that that didn't come really, that didn't come forth properly, Luke. Very early on, Chaitanya worked with phospholipids.
Yeah, sorry.
Yeah.
Yeah, sorry. Sudha, it was an early question before I saw all the more recent stuff you do.
Okay.
Yeah.
So, I mean, we really understand that, see, even though some people, particularly people like Yutetsu Kuruma, who was at LC and who's moved on now, claims that, you know, phospholipids would have been readily formed under early Earth conditions. And a couple of, two pieces of work have actually shown that it's plausible, but it's still very difficult. The more readily available amphiphile would have been fatty acids. And so it's pertinent for us to be working with them. And that is what we did. We went through this after Chaitanya helped setting up the lab and trying to get some of the initial experiments going. One of the first things we did is to veer away from the phospholipid systems. We just use it for certain special experiments, but otherwise we are predominantly working with the more prebiotically relevant fatty acids and fatty acid derivatives. I know Tony now has kind of shifted groups and talks about how coacervates are even more cooler than fatty acid vesicles. But Tony, there was a stage when the protocellular systems did go through this. So yeah, it's super crucial to try and understand these processes using these more realistic, prebiotically realistic amplifiers. And that's something that we've been doing, Luke. And I have to point out here that Luke and we have, Martin, we are collaborating on analog samples from Rotorua. Unfortunately, I do not have much to present because Manish has been very busy trying to get one other project of his done. But now we're going to start focusing on that, on the samples that came from Rotorua, and I'll be very happy to share the results that come out of that in a future meeting.
Amazing. Thank you, Sudha. It's incredible work.
Thank you. Thank you.
Thanks, Sudha. That's fantastic. So if anyone has got other questions that they want to, again, you can feel free to unmute yourself. There's a couple of comments, I guess, Sudha, in the chat, which I don't know if you can see. One from Ian in relation to synthesis of precursor molecules underground.
Which one are you talking about? Sorry.
Sorry, there was just a. I don't know if Ian Miller, if you want to.
Sudha, is it the one? Sorry, is it the one where he says, in my opinion, the chemical synthesis of precursor molecules probably occurred underground?
Yes, that was what I was looking at.
Okay, from my experience, we can get quite a variety under elevated temperature. Polymer sugars and nucleotides would have to occur on the surface. No, I completely can. So I think he's referring to, correct me if I'm wrong, but some of these hydrocarbon molecules form under high pressure, high temperature conditions, like the ones would have been facilitated in the so-called vent systems, right? So yeah, Fischer-Tropsch type synthesis occurs at those kinds of conditions. However, if you want to synthesize your polymers, your sugars, and your nucleobases, you need to have much less harsher conditions, so to speak. So, and more importantly, I think I have to kind of clarify this. I think the hydrothermal vent work is super cool. There's a lot to be learned from that system, and there is some really interesting proto-metabolism related work coming out of those experiments. Nonetheless, one of the concerns I have, or one of the issues that I have with the hydrothermal chemistry is, how do we deal with the dilution problem? A lot of cool chemistry can happen within the vent systems, but as soon as you hit the ocean, yeah, molecules all dissipate. So for that reason, the terrestrial geothermal environment to me is much more readily tangible, in the sense that you have these like Harold alluded to, a lot of interesting clay chemistry is possible in these terrestrial geothermal environments. And depending upon your rock chemistry and other kinds of parameters, the pH can vary, your ionic composition can vary. So the kind of reactions that can be facilitated in these smaller pools, which then are interconnected in some way to some larger pool, you can envisage that multiple prebiotic processes that require specific pH and temperature could have been readily facilitated under these conditions. So yeah, more tangible, more easily can relate more easily to it. Anna says, thanks Sudha, such great work, because the CBC changes a lot with pH. Do you know of any work that relates the apparent pK of fatty acids with the presence of other molecules? So Anna, there's some work, right? I mean, there is precedence to what we have done. What we have shown for the first time is that the C11 system is very cool, and nobody has worked with the C11 system. But having said that, fatty acid alcohol binary mixtures have been looked at extensively. Initially a lot by Dave and group, and then more Pierre has been one of the people that studied this quite a bit, Irene, Sheriff Mansi, and then Kepa. A lot of this work exists. I'll be happy to share some of this. I'll be happy to collate some of this work and share it with you. Yes, pH can completely change the way these systems behave. Tony agrees with my opinion. At some point, membrane-bound compartments definitely were necessary. Yeah, we need to talk still about your coacervates and how sturdy they are under these harsh conditions of early Earth, right? Albert says, I wonder why try ribose. Okay, he's talking to Harold. Mitchell says there's way more to mid-ocean ridge systems than just the highest temperature winds. I would love to know more about them. I'm sorry, I'm just I'm really not clued into those systems very well. I do know that you have these alkaline vent systems, you have these acidic vent systems. The alkaline vent systems heavily propounded by the Mike Russell group of folks. Acidic vent systems much lesser, but yeah, I would love to learn more about that. Ian Miller, I expect the first step from the vesicle is that some peptide synthesis occurs and the peptides get into. Ian, just wait for some more time. We're going to have this really cool paper out. It's in the pipeline. I think we'll be bioarchiving it pretty soon. Sorry. Yeah. I can't hear this person clearly. But yeah, Ian, to kind of, we are not just married to the RNA world hypothesis. We understand that to do that would not be the most appropriate thing to do, because we are completely disregarding the role or the contribution from the other important prebiotic monomers. So one of the things that we're trying to do is to try and understand the crosstalk between amino acids, nucleotides, and amphiphiles. And towards this extent, at least the peptide synthesis part, Manish has done a very neat piece of work which has very interesting implications for messy chemistry and prebiotic complexity, and how when we focus on one kind of reaction, we forget that there are competing reactions that occur that can completely do something not just bad to the main reaction, but something cool too. And this is something we'll put out pretty soon. Rishi, first of all, great talk, thank you. If we go by this theory, what would be the next mechanistic step for nucleic acid to become genetic material for a protocell? And how would one link the broken chains from formation to become? Yeah, so this is something where we'll have to start invoking the RNA to DNA world transition. See, very initially, Rishi, it would have been probably okay for the progenode systems to predominantly use RNA as the genetic material, but the truth is that RNA is less stable than DNA. I mean, RNA does a whole bunch of mind-blowing stuff in extant biology, right? But it's used as the informational molecule only in a handful of entities. And what are they? Viruses. Are they alive? I'm not going to get into that conversation at all. But having said that, viruses are readily able to do this mainly because it gives them a huge advantage. Anytime you throw something at it, they can very quickly mutate and then say, fish, I don't care about what you're throwing at me. I can now mutate and I can still infect, right? So there is this advantage of having a molecule which can encode information, but shoddily. But then the complexity that is required, informational, in order to evolve into more complex forms, one has to be able to store information much more strictly. You can't have a moiety which is so readily mutatable, right? And that is where this transition from RNA to DNA world would have been imminent. And now we've already talked earlier in the day about Ram's work on these RNA DNA hybrids, and there might have been this intermediate world where you had both these molecules, and eventually DNA got relegated to doing what it's doing while RNA does what it does, and proteins essentially the workhorses of the cell. I hope that's answered your question to some extent. Anisha, thank you again, kind words. You talked about polymerization of activated nucleotides. Is there any evidence that these activated nucleotides were, yeah, so a lot of work, 2 decades, a decade and a half of work. I mean, it continues even today, maybe 2-3 decades of non-enzymatic polymerization work is fundamentally based on using activated nucleotides. Are they cool? They're super cool. Can my PhD students get out with a degree within a reasonable time span? Absolutely. Having said that, why do we not work with that? Extensively, we work with that. And that is, I mean, full disclosure, we do work with that, but in certain contexts, right? It allows you to understand processes in a much more cleaner way. So you understand how you can go from A to B without having to understand why C formed, why D formed, why E formed, and what is it doing in the process. But having said that, the prebiotic conditions under which they can form are still not, okay, the conditions under which they can form are clear, but whether they're prebiotically relevant is heavily debated. And where can you find a concentrated pool of these molecules readily available to undergo these reactions? super unclear why these molecules, as much as they can go this way, forward direction reactions, as much as they can readily sort of polymerize, they can readily break down. You veer away from a comfortable pH, your activated entities are all gone. So then the question is, should we start thinking about working with more realistic scenarios? Yes, difficult, but try and understand how this might have happened. So that is where we are at. Anisha, Helen says that, Anisha, I heard Sudha say that her work with ordinary nucleotides was a better model. Helen, I'm not sure I said it was a better model. I said it was a more prebiotically relevant model. I think in science, there's nothing better than the other, right? One is always trying to understand things from diverse perspectives. I just think what we are trying to do and what Dave has always sort of heavily sort of canvassed for is to move to more prebiotically realistic scenarios. Yes, when we do that, things are difficult, things get extremely messy, and that is where I'm aligned more with some of my colleagues from LC who talk a lot about messy chemistry. But one shouldn't shy away from the messiness, because that understanding that will completely change the way we think about how we went from chemistry to biology on the early Earth. That's all.
Excellent. Thanks, Sudha. One more down the bottom. We've got a couple more minutes. Is there anything you've found from the rock records? If not, then what could be the approaches?
More readily answered by Tara, because we don't work with rock records, Prebrata. So we just totally rely on the work that geologists do to try and understand what the rock records are saying, and work from Tara and Martin has shown that these hot spring deposits, which are really, really, really old, 3.85 billion years and older, actually might have some of the first clues of living systems on the early Earth. So yeah, I mean, stromatolites, Pilbara stromatolites, which was initially the rock star example, showed that life forms are cyanobacterial kind of life forms did exist about 3.5+ billion years ago. So yes, there are rock records, if that is the question.
Excellent. We're a couple of minutes before 3, but if unless people have got further questions, we can always just leave it in the chat, and we can wrap up this session a couple of minutes early to give people a bit of an afternoon break before we hit again at 3:30 to continue on with our messy chemistry story. But I just want to thank all of the speakers in this session, particularly Sudha here at the end. Really, really good discussions happening. And we'll take about a half an hour break or something now, and we will reconvene at about 3:30, wherever you happen to be sitting on this pale blue dot. All right, thanks all, and we'll catch you again in about half an hour or so. Thanks, Sudha.